Imagine you are a farmer. You've spent months, tending to your fields, getting everything perfectly dialed in for the season.
Yeah, it's a huge investment of time.
Exactly. And it's late spring, right?
Yeah.
The weather has been beautiful. But then, out of nowhere, you get this brief, seemingly harmless 3-day drop in temperature.
Right.
And we're not talking about a deep freeze here, just down to about 12 degrees Celsius at night.
Which doesn't sound that bad.
No, it doesn't. You walk the fields the next morning and the plants look absolutely fine. I mean, the leaves are green. Everything seems totally normal.
Mm-hmm.
But then weeks later, it's harvest time, and your crop yield has mysteriously dropped by over 50%.
And that is just the ultimate agricultural heartbreak. Because what you're dealing with there is an entirely invisible threat.
Yeah.
It's this silent catastrophe happening on a microscopic level, and it specifically targets the plant's reproductive system.
And today, we're going to expose that invisible threat, which biologists call cold-induced pollen abortion.
Yeah.
Welcome to our deep dive. We are cracking open a massive, newly published paper from the journal Nature. This is dated June 2026, and it's titled, Cold-Induced Peptide Signaling Secures Pollen Resilience and Crop Yield.
It's a fantastic paper.
It really is. The goal here for us today is to get into the exact gear-by-gear molecular machinery that plants use to, sense the cold and save their pollen. We're going to leave no stone unturned. We're walking through the genetics, the cellular alarms, the chemical floodgates, all of it.
Right, and the overarching revelation in this research is that plants aren't just, sitting there passively taking damage when the temperature drops.
They fight back.
Exactly. They actually have this deeply hidden, incredibly complex biological algorithm designed to fight back.
Which is wild. I'm trying to picture how a plant even manages this. I tend to think of a plant's reproductive system as a high-stakes manufacturing plant.
Oh, that's a great way to look at it.
Yeah, where the supply chain has to be perfectly timed. If one delivery of nutrients is late, the entire production line of pollen just shuts down permanently.
Mm-hmm.
So where does this breakdown actually start when the temperature drops?
Well, the researchers started by looking at phenotypic and cellular evidence, basically establishing very strict thermal thresholds. They used a model tomato plant for this.
Okay.
Specifically, it's the Solanum lycopersicum cultivar Ailsa Craig.
Ailsa Craig, got it.
Yeah, and when that specific tomato plant is kept at an optimal 25 degrees Celsius, the fruit setting rate is a really healthy 64%.
Nice.
But if the night temperatures drop to 12 degrees Celsius for just 5 days, that yield plummets to 31%.
Wow. Cut in half from just 12 degrees.
Right, and if you drop it to 8 degrees, it's catastrophic. The pollen is just almost entirely wiped out.
Wait, but the timing of that cold snap has to matter.
Right.
If we stick with the manufacturing plant analogy, there are different stages of production.
Oh, absolutely. Pollen development is highly regimented. It goes through very, very specific stages.
Okay.
It starts at the microspore mother cell, or MMC stage.
The MMC stage.
Right. Then it moves to the tetrad stage, then unenucleate, binucleate, and finally you get mature pollen.
Okay, so a clear assembly line.
Exactly. And what the data shows is that the MMC stage is hypersensitive. If a cold snap hits right then, the plant simply cannot recover.
It's just game over.
Pretty much. However, if the cold hits slightly later, during the tetrad to unenucleate stages, the pollen survives.
Oh, really?
Yeah, it possesses this hidden resilience mechanism.
Okay, so if the pollen itself is freezing, who is actually stepping in to save it? Is there a specific cell doing the heavy lifting here?
Yes. The survival of the pollen entirely depends on this surrounding layer of specialized nurse cells, and they are called the tapetum.
The tapetum. Okay, let me try to visualize this. It sounds like a combination of a catering company and a construction crew.
I love that.
Yeah. Because its entire job is to surround the developing pollen, right? It delivers the essential nutrients, and it also provides this really tough material called sporopollenin to physically build the pollen wall.
That is spot on, and the core conflict of this entire biological drama centers right on that tapetum.
How so?
Well, for the tapetum to actually do its job and deliver all those materials, it must die.
Wait, what?
Yeah, it is programmed to undergo programmed cell death, or PCD, at an exact microscopic moment. The cell literally breaks down and all those nutrients and building materials are released onto the pollen.
Oh, I see.
But the problem is that chilling stress actively delays this programmed cell death.
Okay, let me pause you there. So it's like a time safe full of food.
Right.
The pollen is on the outside, waiting for the food, but the cold essentially freezes the timer on the safe, so it doesn't open.
Exactly. The tapetum stays totally intact, and the pollen literally starves to death right next to a locked-up pile of nutrients.
That is just tragic, starving next to a buffet.
It really is. And if the tapetum remnants persist all the way into the binucleate stage, the pollen aborts. So the plant's survival depends on forcing that delayed programmed cell death to happen anyway, basically overriding the cold's delay.
Okay, let's do a really quick 5-minute recap for you listening just to make sure we've got the foundation solid.
Good idea.
We have a tomato plant. A 12-degree cold snap hits. This cold freezes the timer on our tapetum nerve cells, stopping them from dying and releasing their food. And the pollen is about to starve.
Perfectly summarized.
So the critical vulnerability here is pure timing. But if you're listening to this, you're probably wondering the exact same thing I am. How does a plant, which has no nervous system, actually feel that it is 12 degrees outside so it can try and force that safe open?
That was the big question. So the researchers hunted for the plant's internal thermometer using transcriptomics.
Transcriptomics, meaning looking at the RNA.
Yes, exactly. They were looking at the RNA to see which genes the plant suddenly turns on when it gets cold, and they were specifically searching for small signaling peptides, or SSPs.
SSPs, and what are those exactly?
They are essentially tiny protein messengers.
But the genome is massive, right? They must have been sifting through an absolute ocean of these peptides.
Oh, they were. They started with over 1,100 SSPs.
Wow.
And they filtered that massive list down to 127 that are totally silent under normal sunny conditions.
Okay, narrowing it down.
Right. And out of that extremely narrowed list, they found two crucial genes, SlRGF9 and SlRGF10.
SlRGF9 and 10, meaning these two genes are doing absolutely nothing when it's a nice 25 degrees out.
Zero expression. But the very moment the cold snap hits, these genes heavily express in the tapetum, starting exactly at that vulnerable MMC stage and peaking at the tetrad stage.
That's incredibly precise timing.
It is. And to prove these were the actual emergency responders, the researchers used CRISPR-Cas9 genetic editing to knock them out entirely.
Ah, the classic biological proof. If you want to know what a part in a car does, take it out and see if the car still drives.
Exactly. So they created single mutants, meaning plants missing either SlRGF9 or SlRGF10, and a double mutant missing both.
Let me guess. At 25 degrees, those mutant plants look perfectly fine because the genes weren't needed anyway.
You got it. But at 12 degrees, the double mutant suffered a massive 50% drop in pollen viability compared to a normal plant.
50%?
Yeah, because the tapetum just wouldn't die on time. Its remnants persisted all the way to the binucleate stage.
Okay, here is the biological detail I found super fascinating in the paper. These specific peptides have a feature called a dy motif.
Right.
And for them to actually function as a messenger, they have to undergo this post-translational modification. The plant uses a process called tyrosine sulfation. Can you explain that?
Sure. So the plant physically attaches a sulfate group to the tyrosine amino acid within that dy motif.
Just sticking the sulfate group right on there.
Yeah, exactly. And adding that sulfate group fundamentally changes the physical shape and the electrical charge of the peptide.
So these sulfated peptides are our emergency flares.
That's a great analogy.
They're completely hidden in the plant's genome, doing absolutely nothing. But the second the temperature drops at 12 degrees, they're modified, armed with a sulfate group, and just launched into the cellular space between the cells.
But, you know, a flare is totally useless without someone looking at the sky.
Right. The flare is just floating around in microscopic space, doing nothing unless it's seen.
Exactly.
Let's do another quick structural check of where we are for the listener. The cold threatens to starve the pollen by locking up the tapetum. The plant senses this and fires off these armed sulfated emergency flares, SlRGF9 and 10, into the void.
Right.
So pose this question to yourself. What exactly is the biological eye that sees these flares?
Well, to find that eye, the researchers had to move from genetic evidence to biochemical and structural evidence. They needed to find the receivers on the cell surface. So they searched through a massive family of proteins called leucine-rich repeat receptor-like kinases.
Okay, LRRRLKs. That is some serious scientific alphabet soup right there.
I know.
Help me out with what that actually means in plain English.
Okay, think of them as specialized antenna proteins stuck right through the cell membrane.
Antennas, got it.
They specifically searched the XI subfamily of these antennas, and the winner was a receptor named SlRGF6.
SlRGF6.
Yes, and the structural fit between the flare and this antenna is molecular lock and key on just a stunning level. SlRGF6 has two highly specific structural pockets.
Right, and I actually have the motifs written down here because they're so specific.
Yeah.
It has an RXR motif, which physically binds to the tail end of the peptide, the C-terminal aspartate or histidine, using specific amino acid residues designated as R463 and R465.
Very precise.
And then it has a second pocket, the RxGG motif, that specifically recognizes and binds to that armed sulfated tyrosine we just talked about. And it uses residues R200, G202, and G203 to do that.
Exactly. And here's the kicker. If that sulfate group isn't attached to the flare, the peptide literally cannot fit into the RxGG pocket.
It just bounces off.
Right. The researchers proved this using isothermal titration calorimetry, or ITC.
Wait, back up. Isothermal what?
Isothermal titration calorimetry.
Okay.
It is this highly sensitive technique that measures the microscopic heat released or absorbed when two molecules bind together.
Oh, that's clever.
Yeah, and by measuring that tiny bit of heat, they can calculate the dissociation constant, known as the KD value.
And what does the KD value tell us?
Well, a lower KD value means a tighter, stronger physical group. So the KD was 258 nanomolar for RGF10 and 676 nanomolar for RGF9.
I'm assuming in the world of molecular biology, nanomolar ranges mean this isn't just random cellular bumping around. This is a very powerful magnetic attraction.
It is a definitive direct physical bond, but, and this is a big but, the receptor SlRGF6 can't act alone to sound the alarm.
It needs help.
Yes. It requires co-receptors to physically confirm the signal. Enter the SlRG family of proteins, specifically SlRG1, SlRG3A, and SlRG3B.
Okay, so we've got SlRG proteins now.
Right. When the RGF flare binds to SlRGF6, the peptide acts like a molecular glue. It forces the main receptor to grab and hug one of these SlRG co-receptors. We call this heterodimerization.
Heterodimerization. So SlRGF6 is the lock, and the SlRG co-receptor is, like a deadbolt?
Basically.
So the plant is essentially running a highly secure, multi-factor authentication system.
I love that analogy.
A main receptor is the lock, SlRG is the deadbolt, and the RGF peptide is the highly specific sulfated key. If any piece of that is missing, access is denied.
Which is exactly what they found. When the researchers mutated the plants to lack the SlRG3A and 3B co-receptors, the whole rescue mission totally failed.
Even with the flares.
Even if the flares and the main receptor were present, without the SlRG co-receptors to complete the hug, the signal just died right there on the surface.
Let me lock in this step of the process for everyone. We've got the emergency flares drifting through the cellular space. They perfectly slot into the SlRGF6 receptor on the outside of the cell,
Yep.
but only if they're sulfated.
Right.
Then, the SlRG co-receptor comes over to verify, completing our multi-factor authentication.
Yes.
But all of this is happening on the outside of the cell membrane. So if you're tracking with us, the big question is, once the lock turns on the surface, how does the message actually get inside the cell to trigger the timed death of the tapetum?
Right. We have to follow the signal inward.
Okay, take us inside.
So transcriptomics showed that cold fundamentally alters the expression of calcium channels in the plant.
Calcium channels.
Specifically a type called cyclic nucleotide-gated channels, or CNGCs. And the crucial players here are SLCNGC16 and SLCNGC18.
So we are connecting the antenna on the surface to these calcium channels deep in the membrane.
Exactly. Let's trace the exact chain reaction. Once that multi-factor authentication happens and the SlRG co-receptor hugs SlRGF6, this SlRGF6 receptor undergoes autophosphorylation.
Autophosphorylation.
Yes, it actually reaches around and adds a phosphate group to a very specific spot on its own structure.
Oh, wow.
An amino acid called lysine 797.
And adding a phosphate group is basically biology's way of flipping an on switch, right?
That's a perfect way to describe it.
And we know this spot is critical because when researchers mutated just that single amino acid changing the lysine to a glutamate, which is a mutation called K797E, the entire multi-million-year-old system completely broke down.
Yep. Changing that one single building block ruins the switch.
Incredible.
And once it's successfully activated at lysine 797, the receptor complex reaches down inside the cell and directly phosphorylates the inner tails, the N and D termini of those SlCNGC16 and 18 channels.
And this phosphorylation literally rips the channels open.
It does.
The researchers actually watched this happen live, which is so cool. They used a fluorescent calcium sensor called GCAM-P6 meter.
Right.
And the moment those channels get phosphorylated and open, there's a massive influx of calcium rushing into the cell, and the sensor just lights up.
That intense calcium spike is the final undeniable signal. The calcium flood is what finally triggers the programmed cell death of the tapetum.
And they proved this, right?
They did. They proved this conclusively by applying a chemical called lanthanum chloride, or LCL3.
Because lanthanum chloride physically blocks calcium channels from operating. Right?
Exactly. When they applied it, the calcium couldn't get in, the tapetum didn't break down, and the pollen rescue mission failed entirely.
This visual is just incredible. The receptors on the surface are like the security guards checking IDs.
Yeah.
Once the sulfated key is verified, they radio down to the actual physical floodgates, the CNGC calcium channels, and order them to open. Calcium rushes in like a cellular demolition crew to take down the tapetum so the pollen can finally eat.
It is a breathtakingly coordinated mechanism, and all of it just to survive a few nights at 12 degrees.
Okay, let's step back and look at the blueprint we've just mapped out in this last segment.
Good idea.
The cold threatens the plant. The plant fires the RGF flares. The flares hit the multi-factor authentication receptors on the cell surface. The receptors flip their internal switch at lysine 797, which rips open the calcium floodgates.
Right.
The calcium rushes in and destroys the tapetum, saving the pollen. Now that we had this entire schematic, the really obvious question for you listening is, how do we hack this algorithm to protect our global food supply?
And that translation from lab discovery to actual agronomic application is where this gets absolutely critical.
Yeah.
The researchers theorized, well, if RGF9 and 10 are the emergency flares, what if we just permanently give the plant more flares?
Okay.
So they used a genetic tool called a 35S promoter to artificially overexpress the SlRGF9 and SlRGF10 genes in tomatoes.
A 35S promoter. So that is essentially swapping out a standard light switch for an always-on mechanism, correct? You are forcing the plant to just pump out these peptides continuously.
Yes, but they discovered a really crucial nuance in the field, the Goldilocks effect.
Not too hot, not too cold.
Exactly. When they generated lines with moderate overexpression, specifically the transgenic lines 35S SlRGF9 1 and 10 1, it was a massive success. It prevented cold-induced yield losses by up to 52%.
52% yield recovery.
Huge.
I mean, if you are a farmer facing a cold snap, that is literally the difference between bankruptcy and a profitable season.
Absolutely. It's game-changing. However, when they pushed the system too far and created lines with excessive overexpression, it backfired entirely.
Oh no, what happened?
The plants suffered stunted growth and even became completely sterile, so the dosage of these signaling peptides must be incredibly precise.
It makes total sense, though. I mean, if you sound the emergency alarm at maximum volume all day, every day, the plant burns all its energy, panicking when there's no actual threat.
Exactly.
It has to be perfectly tuned. But, you know, the ultimate aha moment for me reading this paper was realizing this isn't just a tomato cork.
Oh, not at all. The researchers looked across the plant kingdom and found that this exact same molecular algorithm exists in monocots too, like rice. They identified the genetic orthologs in rice, OSRGF8 and OSRGF10.
And for anyone wondering, an ortholog is just a gene in a different species that evolved from a common ancestor and retains the same function.
Right, and they actually took it out of the greenhouse and into the real world.
Yeah, the field trials.
Yes. They ran major field trials in Beijing and Yunnan using a highly commercial, top-tier japonica rice variety called Zonkefa 5 or ZKF5.
And the results?
By moderately upregulating OSRGF10 in this commercial rice, the plants were able to recover 18.3% of their grain yield loss under chilling stress of 15 to 17 degrees Celsius.
Man, when you multiply 18% across millions of hectares of rice paddies globally, that translates to an enormous amount of secured food supply.
It really does. It's a massive scale.
So we've seen that hacking this hidden network works, and it translates directly to agricultural yield recovery in both tomatoes and rice, provided the dosage is strictly moderated. Let's synthesize this entire molecular journey one last time to ensure we have the whole picture before we go.
Okay, the chronological path is vital here. It starts with the farmer's field dropping to 12 degrees Celsius.
Right.
That chilling stress threatens to delay the programmed cell death of the tapetum, which would leave the pollen to starve.
But the plant pushes back. It launches its hidden SlRGF9 and 10 emergency flares, ensuring they are armed by that tyrosine sulfation.
And those flares drift to the cell surface and bind to the SlRGFR6 and SRC locks, completing our multi-factor authentication.
The receptor flips its internal switch, autophosphorylating at lysine 7-9T7.
Which then reaches down and physically opens the SLCNGC16 and 18 floodgates.
Calcium rushes in like a demolition crew.
Forcing the tapetum to break down exactly on schedule.
And the pollen is rescued. The crop yield is saved.
It's a beautiful system. And looking forward, the implications of this June 2026 paper go far beyond just mapping a cool mechanism.
Definitely.
We are facing unprecedented shifts due to climate change. Agricultural zones are migrating rapidly.
We are seeing a massive increase in unpredictable weather patterns. Specifically things like false spring cold snaps, right?
Right.
Which gets unseasonably warm. The plants wake up and start flowering, and then the cold violently returns and just devastates them.
Exactly. And the paper notes that instead of using blunt genetic tools like the traditional 35S promoter, which risks that Goldilocks burnout we talked about, we can now use advanced synthetic biology, specifically tools like prime editing.
So instead of just permanently flooding the plant with flares, prime editing lets us act like a sound engineer tweaking a mixing board.
That's a great way to picture it.
We can gently edit the promoter regions of the DNA itself, essentially adjusting the sensitivity dials of these specific genes. We could teach the crop to launch the flares just a little earlier, or maybe a little more efficiently, perfectly tailoring it to whatever its new changing environment requires.
And our ability to locate and activate these cryptic, dormant resilience networks might literally be the key to averting global famine in the coming decades. It fundamentally changes the paradigm of agricultural science.
It really does. It completely changes how you look at a field of crops out your window. They aren't just sitting there passively enduring the weather. They are actively calculating, defending, and waiting to deploy these brilliant microscopic mechanisms that we are only just beginning to decode.
It's humbling, honestly.
It makes you wonder, if this stress response network was completely hidden under normal conditions, how many other invisible protective systems, just as elegant as this calcium floodgate, are sitting totally dormant in the DNA of the staple foods we rely on every single day?
A lot to think about.
Absolutely. Thank you for joining us on this incredibly rigorous exploration today. Next time you feel a sudden chill in the air, remember the microscopic demolition crews springing into action. Keep questioning, keep digging, and we will catch you on the next deep dive.